will say something about that later on to introduce that topic. Because we first want to listen to Ricard Solé, and I invited Ricard to join us also to give a little bit a different perspective on the kind of phenomena we look at, because so we often think about these high-level phenomena that we looked at last week, like cognition, behavior, problem-solving, invertebrates, from a pure neural perspective. So you always think about, okay, whenever something interesting happens in nature, it has to, in the end, be done by some neural structure or a brain-like structure. But in some sense, you can think of living systems in a much broader perspective, in a broader perspective of what you also could call dynamical systems. And I thought Ricard would be the right person to get us straight on that track, to just give it this idea. Look, if you want, you could also argue there are more ways to skin a cat, right? There are more perspectives on living systems and behavior than only one that is sort of very neurocentric. So Ricard has a really broad experience in this field, and it's really in the kind of work Ricard is doing, which is absolutely outstanding in this field. I think he's done an amazing job in bringing together a more formal understanding of really complex biological systems, and both from an analytic and a synthetic perspective. So that means it's also a bit this methodological issue we discussed last week, that it's not only about decomposing systems and trying to get some description of the pieces, but it's also about putting all these pieces together in a scientific framework or in a mathematical framework to get an understanding of underlying principles, because in the end it is all about underlying principles. So Ricard is also the Cray professor here in Barcelona. Has a fantastic track record that will reflect in every slide we're going to look at. And I'm really happy that he could join us. And actually we're in a time machine now, because today is the 2nd of September. So Ricard, thank you very much for joining us. I'm looking forward to your talk.
Okay. You guys hear me?
I don't, yeah, that should be working, yeah.
We're recording the lecture.
Oh, okay. You hear me now.
If it's okay.
Very good. Well, thanks for the invitation. I'm sure my talk is going to be non-standard. That's the usual thing that tends to happen. My group has been working over the years in a really broad number of issues. So if you can put the lights a bit down. If I had to say in a short number of sentences, what we are looking for would be something like maybe the general laws of complex systems. Are there laws somewhere? And that's the reason in the title, in the original title, why I had the logic of monsters as a kind of as a subtitle. As a scientist, you look for regularities in nature. You look for laws, and you could formulate that in a completely different way, apparently, which is the following. You know that in the old times when the explorers reached the end of the world, they went back saying that they have seen monsters. And as time unfolded, what happened is that as the world became more and more known, the monsters became less and less common until they completely disappeared. They inhabited these blank spaces in the maps, and when the blank spaces vanished, the monsters vanished. But there's a question that remains there, essentially, which is why there are no monsters? So why, when we observe nature, there are things that we don't observe, okay? For example, in this picture, I illustrate the concept of the monster. Why no monsters like this? And I'm referring to the one at right, not the one at left. Why no lions with wings? And that might sound like a strange question, but it is not. The fact that there are some things that are not present can tell us something about why we do observe regularities to see in nature. In recent years, something has happened I could say in biology, but it's much more general, I think, in terms of the approach that we make when we try to understand, in particular, biological systems. And that's related with an area which I can label as artificial life. where many questions arise from what's the origin of life, from what life is actually, to the question of what is life and can we imitate life in any way, right? Some of you, whether or not you're aware of that, are doing that. Through experiments and observations, we try to understand biology. Theory and simulation are the other two legs that we use to approach biological complexity. But in the recent years, we have a new area that doesn't fit any of the previous ones. Synthetic biology, which I try to illustrate with examples, is a field where essentially we use techniques from genetic engineering on a scale that is unprecedented to manipulate life, to build things that do not exist in nature. And by doing that, we have been able to find out that a number of things that we assumed on how the evolution has shaped aging, death, all kinds of things, and what's the impact of genetics on a number of issues from cancer to reprogramming life, were completely wrong. So we can manipulate things and build things that do not exist. And why synthetic biology is not experiment? Because you can really very much plan what you want to do. You can construct things that are not part of nature, and you can simulate that before. So it's in the middle of nowhere, really. Let me do some little historical background. Artificial life, the idea that we can imitate life is quite old. And in the 19th century, automata, mechanical automata, were kind of really the big fashion thing that people was doing. This is an automaton by Jacques Droz that still exists. It's in a museum. So pretty much in contrast with any iPad or wherever you can imagine that in 2 years it's already old-fashioned. This still works. Automata were the big thing of the time and were very interesting for many reasons, including sociological ones. There's a lot of funny stories about this automata. For example, the inventor of this automaton have several inventions. One of the automata was capable of writing very, very nicely this sentence by Descartes. I am, so I exist, or whatever it is, in translation to English. So they went to Spain in tour, which was a bad decision because Spain at the time was quite a nice place for the Inquisition. And building an automaton that imitating a human being was considered as a heresy. So the funny thing is that the builder and the automaton went to jail altogether for a couple of days. So if you think about it, it's funny because actually the people who actually considered that kind of a heresy gave the automaton the status of a human. So anyway, the most famous of all the automata was the Turk. The name you see quite well. Why is that? The Turk was a very impressive machine, human-like, the size of a human. A lot of mechanisms, mechanical mechanisms, were at the bottom shown before the acting by the builder. There was a lot of theater in the presentation. It was very impressive. Anyone who played with the automaton, which lived kind of 94 years, I think, was quite much impressed. He won, or it won, almost everyone, playing with a lot of people, including Napoleon, who it is said when he lost the play, he was really, really angry. He played with all sorts of people and scientists. Charles Babbage, who you know was the inventor of the first mechanical computer, played with automaton, was very, very impressed. He was kind of dubious about that. Was it possible that an automaton was able to play 19th century chess, really? But he had a lot of influence. You see that he says, in describing his play, he played very cautiously, like human-like. And despite that he had a lot of doubts, it was a big influence in his career because he kept thinking, maybe I could build not only a machine that calculates, maybe a machine that thinks. So what happened with that? Was the Turk really a machine? Unfortunately not. It was really, really a a big fraud. And several people tried to unveil the fraud. Edgar Allan Poe was one of them. He went to watch the automaton several times when he was playing in Baltimore and wrote this essay that I strongly recommend. It's completely devastating. It's a kind of a scientific analysis of the play, showing that for several reasons the automaton had to hide someone inside, which was, of course, what happened. The automaton over the years was hiding different people, very good players, not dwarfs, just normal people, because the whole thing was really a big invention of how to put a person inside that. And the end of the automaton was spectacular. It was like in a movie. It was sold to someone at the end of their life. Ended up in a museum in Boston, but the museum got in flames. It was on fire, and the owner tried to get the automaton, to catch the automaton or save it if you want, and the automaton was burning. And I forget to say the automaton was able to say some things like chess and chessmate. So the automaton was burning and saying chessmate, chessmate, which was very appropriate, I think. So if you look in historically in the term artificial life, you can actually scan that by using information that is in the internet. I don't know if you are familiar with the Google Ngrams engine. Surprisingly, not many people knows about it. It's a very simple idea, very powerful, and it's a bit addictive. Just a warning of that. Google has digitalized millions of books, and so it's a database you can enter. You put a word, or 2 words, or 3 words, whatever. For example, artificial life, and gives you a scan on the whole collection of books at the time, how often this term was used. Okay, so it's interesting because it provides a kind of a picture of the relevance of the term over time. And artificial life has this bit peculiar profile. You have this sharp growth in the early 19th century, then a decay, but it never fades out, as many works do, and then it gets back again in the 70s. Any guess about the first peak? First peak is Frankenstein. Yeah. It's Mary Shelley. Mary Shelley, she publishes 18 years old, which is not bad at all. Publishes Frankenstein, goes up, goes down, but never fades. And in the 70s, genetic engineering comes, and to some extent, artificial life becomes really possible. So one of the main threads that connects the things I'm going to talk about is computation. We use computation as not only a technical form for approaching systems, but also, as John Hopfield raised in this very interesting paper, that computation is actually something that makes biology different from physics. Anything in biology, from molecules inside the cell to organisms, even populations, perform computations, gather information from the outside, process that information, and respond to that. Okay? Everything in biology works like that. And computation implicitly or explicitly is there. And sometimes in ways that we don't realize. For example, you have a lot of cell types inside your body. So types of cells that are specifically well defined by genes that are essentially on or off. Okay? And that's a very simple computation. You have switches that, through development and within your life, work well to provide an organized structure. When they don't work well, as many diseases, that's something I tell my students, that it's an example of when computation doesn't work well. For example, this is from the guy where cells that should differentiate into muscle differentiate into bone cells. So that's a terrible disease, and there are other that have no cure yet. So the idea that cells compute, that molecular systems compute within cells, became more and more commonly accepted in the 90s, when people started to see that some of the circuitry inside cells reminds us circuits as engineers design. And now, as I was saying before, we jump into one possibility that was unexpected years ago, which is not just looking at the circuits or modeling, but building them, all right? That's synthetic biology. When journalists come to my lab to ask about that, they inevitably end up talking about Frankenstein. So as it's said here, if you cannot win them, you join them. So what is synthetic biology? This should be a movie of Frankenstein, but unfortunately it's a black box. So, but you guys are working in black boxes most of the time, isn't it? Well, this should be a scene of Frankenstein anyway. Interestingly, in Frankenstein, there is something that is truly what we do, not unburied bodies, dead bodies, but Victor Frankenstein at some point says something that is quite modern. If you want to become really a man of science, you have to really be an interdisciplinary kind of guy, and should advise you to apply to every branch of natural philosophy, including mathematics. These dots there are very insulting comments about experimentalists that I tend to remove. It might sound strange that mathematics can tell us something about biology, especially when you think about very formal models of mathematics. I just want to point an example that I think is really, really interesting for a number of reasons. From the logic of life to the logic of monsters that I was mentioning at the beginning. For Neumann, one of the greatest mathematicians of the 20th century, and probably the last Renaissance mathematician. At the end of his life, for Neumann, he was involved in lots of areas in mathematics, originated game theory, had big contributions to quantum mechanics, and at the end of his life, he was very, very interested in biology. So how do you approach biology from mathematics? And he was interested in a number of really, really fundamental questions in the 50s. And one of them was, what is the requirement that they need to have a machine that self-replicates itself? I'm talking about years ahead, molecular biology, years ahead, DNA, polymerases, proteins, or anything about the logic of real biology. And to make the long story short, he ended up in a basic scheme, which essentially says that the machine has to have a set of instructions, and the instructions have to define how to replicate the machine. But when you replicate the machine, the instructions have to be replicated also. And the basic building blocks of that, which he called this duplicator, the controller, etc. If you go to a molecular biologist today, he will say, oh yes, the polymerase, the regulatory network that provides the machinery to control the process, etc., etc. Suggesting that maybe life, as we know it, is unique. So it's only unique, but not unique in terms of a kind of an impossible thing. But the only way of organizing a system that self-replicates is what we already know. So synthetic biology comes in the last 12 years, maybe, as the daughter of genetic engineering, with a really powerful set of tools to modify life. One paper that you might have heard about appeared, I think, 3 years ago by Benter's group, involved an example of what can be done. Spectacular in terms of the engineering. Essentially, these people, what they did was to build a whole genome using computers. So it's a chemical construction of a genome, which is in the end is a copy of a genome that was known, except that you can manipulate a number of things inside. They put this synthetic genome inside the cell that was from another species, which previously was removed, the whole genetic material, and they kind of reboot the computer. What happened? But the cell started to work again. Well, this is an interesting event. Craig Venter is a kind of peculiar guy who tends to oversell his work. But it is true that they did something unique, and which is that until then, every cell in our planet was part of a chain of events that connects us from the ancestor. Every cell came from another cell. That's no longer true. That's no longer true because that's what happened. Incidentally, I found quite kind of funny a comment that Venter did after, I think it was a TED talk, when a journalist asked him, people is saying that you guys are playing God. And he said, who says we are playing? So, synthetic biology is still in the infancy. So we are now, I think we are entering the exponential phase. A number of things have been done. So you can manipulate genes and build circuits, which means that you can think in the logic of something that you want to build. For example, a switch. You have two different states, and you want to have the system reaching one state or another in a stable and controllable way. And the thing that makes us free is that you can use the building blocks that you use, the genes, the promoters, the proteins, and everything can come from any species. So you can use, we use, for example, in our lab, promoters from a virus, gene from a bacteria, another gene maybe from a mammal cell, what is at stake and works. And so you can build switches, a very important part. You can build clocks, for example, oscillators in a controllable and tunable way. People has made proofs of concept of a number of things. You can build, for example, using a plate, a petri dish on a two-dimensional setup, a system made by bacteria that detect the boundaries of a picture, all right? And one of the big things that might happen with the whole story of synthetic biology is the potential for approaching disease in a completely new way. In most diseases are so-called complex, so it's not like a single element that fails. It's a part of the whole network that is built in our organism that fails. So it's a regulatory machine that doesn't work. So could we restore the function even when this functionality is destroyed by your own organism? I'll illustrate that with an example later on. By rebuilding the circuit, being able to do it as people has done, using cells that do not perform that function, okay? And the answer seems to be yes. So going into computation again, because computing in the end is almost everything, even what I just mentioned about a disease, the circuitry that fails is a computation that fails. So can we restore that? Is it possible to engineer a living computer? Okay, and a very important thing if you want to start thinking in general, what are the right metaphors for that? And for that, I'm referring to the theoretical metaphors. Okay, for example, is the Turing machine something that is going to be of help when looking at systems like molecular systems in biology? Well, it is interesting to see that actually, you know that the metaphor used by Turing as a computer is this machine with internal states that reads a tape, okay, very long tape, and that is able to manipulate the symbols in the tape, read and write. Interestingly, several very important machines that you see inside cells, they are not Turing machines, strictly speaking, but they do conform to the scheme of a complex machine with internal states, a macromolecular structure that reads a very long tape, which can be RNA, can be DNA, right? And the result of the computation usually is to build new molecules, such as proteins here in the ribosome, or to perform regulatory functions. So you could say that's not a bad metaphor, okay? Of course, this is not the whole story. There's a lot of things that happen in terms of computations inside cells and in the surface of cells that do not adjust to that. The other part of the engineering metaphor will be going to electronic circuits. And as you probably know, any electronic circuit can be built with logic gates, which is essentially defined by Boolean functions. And in particular, there are a number of gates, so the NAND and the NOR gate, from which it is possible to show that any circuit can be built. So maybe this is a starting point. I just want you to keep in mind two things that I think are relevant, because briefly what we're going to show is that the standard engineering metaphor is not the right one when you're trying to engineer biology. Let's look at a given circuit. I could summarize the idea of what is electronics or this combinatorial design by saying that a given computation can be built by combining logic gates, right, in some given way. There are multiple ways of doing that. That you have a number of inputs, for example, one output, could be more than that. But the thing is that every element in the circuit is connected with the others through some path, and that input and output have this structure. The output is localized in a given place. That seems trivial, and it is trivial somehow. But because it's trivial, it's easy to fall into the belief that this is the way that you have to do things, right? But biology is different, and we have to manage to manipulate life that is inside the cell, and our accessibility, our capacity of manipulating things is limited. So we can build logic gates, for example, an AND gate like this here. This could be kind of a circuit where you have two inputs, which is two input molecules that go and affect two different genes. The genes produce, if they are stimulated, two proteins that go together and then can inhibit another gene, right? You can show that this corresponds to an AND gate. So in principle, it seems that you can do it. But have in mind that the machinery is not like you have in electronics. It's a quite different hardware. So in principle, you could do as in standard combinatorial design. You take two gates, you combine them in different ways, and you have other gates, maybe more complex. So in principle, I can do that inside my cells by engineering properly different genes that I put inside. GFP means green fluorescent protein. In many cases, when we start to building things, the on-off state means that you have the fluorescent signal or not, right? And what happened? Why is not this scheme easy to extrapolate? Well, in electronics, when you build things, what you essentially do is to choose your gates and connect them with wires. And the wires is the same thing. It's a copper or whatever cable. And all the cables are the same. So this, again, is a trivial observation. But when you go inside the cell, the cables are not the same anymore at all. So when I activate a gene and this gene has to send a signal to another gene, I cannot confuse that. It's a specific cable for every pair of things that are connected. Okay? So that might not seem a big thing, but when you try to do it in the lab, it becomes a nightmare as soon as the number of cables go into 2 or 3, right? Not to think about beyond that point. That, for example, will be what in principle will constitute a circuit that is very much used in electronics, the multiplexer, which is easy to build with gates, but it's extremely difficult to build inside one cell. So, in principle, we could do it, but the question is how to do it. And people have been speculating for a long time. Many people, as happens in science, have extrapolated immediately. They build an AND gate and immediately say, well, an AND gate, for example, this paper is a NOR gate. By connecting NOR gates together, any computer circuit can be built. They forgot to say, in parentheses, in principle, which means that it's probably unlikely to happen. So, if we cannot connect logic gates all together in a simple way inside the cell, and we want to build something that we can call a biological computer, how we deal with that? Within cells, besides of the problem of putting different cables, what happens extremely often is that you have responses that you don't want to see, but the cables are shared with different proteins, and we have what is called the crosstalk. So you put a new wire inside the cell, and something that you want to connect, a gene with another gene, ends up connecting other genes altogether, right? So John Wheeler, one of the giants of physics of the 20th century, said, as a kind of an advice to students, which I think is quite good, is in any field, find the strangest thing and then explore it. Okay, I think it's a good recommendation if you are ambitious. So maybe building computations requires a different perspective. Okay, you want to build a living computer. Being a member of the Santa Fe Institute for many years has provided me the privilege to be in contact with people that has contributed very much into rethinking computation. This is Christopher Langton, which was the father of artificial life, thought a lot about what is the dynamical requirements for computation in general in biology. Stephanie Forrest provided the basis for emergent computation, and Melanie Mitchell and Jim Crutchfield, who maybe you know about their work on genetic evolution, simulation of genetic evolution, they thought about what they call emergent computation also in terms of evolutionary responses. So what's the right inspiration? Well, when you look at in nature, there are different ways in which systems perform computations. And ant colonies are, I think, quite a good example of that. They illustrate the idea of emergence. They build really complex nests, and they perform collectively very complex computations, despite that individuals, no matter what you think about it, they're quite stupid, and they interact with quite simple local communication signals. Despite that, they collectively do amazing things. I know every time I show this video, I probably no one is listening to me anymore, but it's there. It's the internet. I can tell you what is this. I have to go into the next slide. Sorry. So a few seconds more. So if you actually want to go into new metaphors, maybe you have to shift some of the assumptions that you made. All right, and that connects with another idea that I think is really, really important. I always insist in that, which is the landscape of potential designs. We tend to think that what is there in engineering and what is there in biology, that's the whole story, and that maybe the landscape is what we observe, and that the landscape is reachable by our mind. What do I mean by that? Imagine that you have a problem that you want to solve. Maybe if you were able to plot the landscape of potential designs, and the peaks in this landscape are good designs that respond to what you want, and the valleys are really bad solutions, a landscape for any complex problem is really complex. We have no really idea about how complex is that. And let me illustrate the idea that I want to raise, that I think that should prevent us from assuming things. Imagine I want to build an antenna that has to be in space. This is something that has been done. And that has to gather information from different sources to analyze the Earth's magnetosphere. And instead of designing by standard design, you make that in a computer. So you use an experiment of natural selection where you start with 100 antennas that are just randomly generated, and you put the antennas under the conditions that you'll find in space, which is physics, and so you can really simulate it quite well. So you select again and again, okay? What is the end result of that? It doesn't seem to be a good way of doing that, okay? And you all have an idea of an image, mental image of how an antenna looks like. Well, the winner of the competition is this, which by any standard, I will say it's unexpected. Maybe some of you guys have seen that before. But the thing is that this is not the outcome of our mind. It can be. We cannot design that, right? It's interesting because that means that it's a peak in the landscape that is reachable by our machines, but not by our minds. So how did we solve that? Because we did solve that. Instead of going into the standard design, so we have cells. Inside the cell, I put a number of engineered things, and I expect that if I combine more and more genes inside the cell, things will work. Let's make three assumptions. The first is not new. Use multicellular systems. So I do not engineer a single cell, but I do engineering in different ways in different cells. Okay, and then I put them together. That's not new. But we made two non-standard assumptions. Okay, and we started from theory, just with paper and pencil. The first is that let's not assume that things are connected. So that maybe my engineered cells define a circuit, but different cells might not be connected with the others, right? Of course, this in electronics is complete nonsense, right? But what happened in biology? And the other thing, let's allow the output, which in a circuit is somewhere very well localized, let's be distributed, which means that different cells that I have engineered can have the output. Not a single one, maybe all of them, right? This sounds bizarre because it is bizarre. But let me illustrate by an example before going into more complex designs. What's the outcome of that? The multiplexer that I just showed you before in a single cell is something that is almost undoable. With our view, with our approach, you can split the multiplexer in two cells. One cell receives two of the inputs, the other receives two inputs, they just have one in common. And one cell and the other can be the output. And as you can see it, they are not connected. There's not a signal going from one cell to the other, right? So it's kind of a circuit that I broke into two pieces, and each piece can be the output. Nonsense in standard electronics, but it works very, very well when you build that in synthetic biology. So we use yeast, which is the simplest cell with a nucleus that you can use as a single entity. And we made a team at the Parc Scientifique, very near the beach. And we extrapolated these ideas that started with paper and pencil in collaboration with experimentalists, so people that could actually do it in the lab. We built a library of cells that today is the biggest one that exists and is really, really well characterized. And we did two things, showing that you can build really complex computations that no one else had achieved before, and you can do it in something that synthetic biology was promising but didn't show up, which is using that in a Lego-like way. So you can use different pieces and recombine them in different ways. For example, using our library, just show a few pieces here. You can build an n-implies gate, a NOR gate, not spectacular, a multiplexer. With yeast, we use three pieces, with three cells, sorry. You see that two of them are connected, but not connected to the third. A comparator, again, you have two cells connected, but not the whole circuit. This works very well, very reliable. It's easy to engineer single cells separately because we don't do a lot of things inside each cell, and you can reuse a lot of stuff. And you can build really, really complex circuits that, if designed in the standard way, either using minimized circuits or just NOR gates or NAND gates, could be really complex, the two examples over there, and that with our method just require 4 cells with little engineering, and that work quite well. So now I'm going to make a list of things that we are doing, that we are asking ourselves, and other people has recently been asking. Particularly because we are interested in trying to push forward that. So the landscape of computation might have other parts that we do not even suspect that are there. One thing that we are interested in is whether or not this distributed computation happens in real biology. Is this something that we have created in the lab but has no counterpart in natural systems? We start to suspect that that's the case. But my colleague at the Santa Fe Institute, Mary Gelman, thinking in terms of quantum mechanics, he said that everything not forbidden is compulsory. The idea is maybe something that can happen in biology ends up happening, which seems to be the case most of the time. One of the big avenues, we're working on that also in collaboration with people that works on the diabetes type 1, which affects children, is, for example, replacing the circuitry of a system that has failed. In this type of diabetes, what happens is that your immune system attacks the cells of the pancreas and destroys them. So the solution cannot be to replace those cells because this is extremely expensive, difficult, and it's just going to last for a short time. So the idea we have in mind is, as other people in the world is thinking these days, maybe you can use cells of your body that do not build insulin, muscle cells, mastocytes, cells of the immune system, engineer those cells and replace the circuit by something that is not going to be attacked by the immune system. It's a big challenge, but it's an extremely fascinating one.
Why is that a computational challenge?
Well, because you have a circuit there. We think that the only way of replacing safely that is by distributing the computations that have failed, which essentially is properly detecting the concentration of insulin and glucagon, which is two things that kind of react in opposite ways. And doing that by putting the pieces in 2 different parts of the body. It's computational in terms of a very simple decision-making circuit. simple, at least simple, written in a paper.
Okay, but you could also argue that you have to sort of change the surface properties of these cells that the immune system, for instance, cannot detect them.
No. If you imagine that, just dreaming about it, that we could be successful and put our production and response to insulin, etc., in cells that are different from the pancreatic cells. So the immune system will not attack them because it's a different story. So that's the idea.
Sure, but why I was posing the question, because you use the word computation quite a bit to describe a fairly diverse set of phenomena, and in some sense we might also be drifting away from a traditional notion of computation, right? So you are redefining what a computation really is, and I wanted to sort of understand that a bit better. Because in what sense is this still, let's say, computation in the Turing sense, for instance, and to what sense is it sort of touching upon new aspects of computation?
Yeah, let me go into the end, maybe, because the last picture might provide maybe a response to that. Other things that we are exploring, I think that successfully, we have our own wet lab, which it's a funny story because we are theoreticians by training for many years, and so when we decided to make a wet lab in our institute, everyone was terrified. The theoreticians going to make experiments. One of the things I have been building is a cellular system, a multicellular system, which has the capacity of associative learning, so they can learn from previous experience, associate signals that initially are not linked to any given response, and show actually that this can be used to interact with a number of potentially biomedical targets. Another aspect of our work and the work of others are doing is reprogramming. So you just very briefly, you know that today it's possible to take a cell of your skin, reprogram that by putting several genes in action into that cell, converting the cell into a stem cell, which is yours. It's not the stem cell of anyone else, it's your stem cell. And then using that cell to rebuild any kind of cell type. That has extraordinary consequences that we still need to understand. 10 years ago, that was completely unthinkable. But the thing is that cells can be reprogrammed, and just to briefly mention that with our method, reprogramming can be made in an extremely flexible way. Very recently, we have, recently, the last 3 years, we've been expanding our picture into what we call spatial computation. We put our distributed system in a setup that is called a microfluidic chip, where cells can live, you can connect them, and you can isolate them in different ways. And by doing that, we've been able to now go a step further from that, building much more complex circuits. We do believe, we have not a proof of that, but we do believe we're really into the limits of what can be done with multicellular systems by limiting the amount of wiring that we can use. And we have built, I think, the most complex things that can be done. Another thing that has happened recently, and that we haven't been involved in that yet, it has to do with engineering organs, engineering tissues. This is a picture from what? What movie? Blade Runner, exactly. In Blade Runner, this is something similarly speculating idea that there's an engineer that builds eyes. You might think, well, it's a movie. But actually, what happens when you put stem cells and you properly cultivate them in a given medium where they are induced into behaving like cells of the eye? What happens is that spontaneously you build eyes. So you have a whole machinery of development that is there, and you prepare the things properly, the eye is formed spontaneously. So you probably heard this summer that something came out. We were very interested theoretically in something like that. What happens if you do that with stem cells that allow you to build tissues of the brain? So you can actually not only create brain cells, which had been done before, but put the things into the right conditions so that the cells have a medium and a substrate where they can actually build tissue. What happens is that you recreate quite well structures that you see in the initial phases of the building of a human brain. So you create parts of the cortex. You can actually manipulate by silencing techniques that are available. You create alterations of that. In the newspaper, of course, some journalists started to talk about the consciousness of the microbrain. I think it's a bit exaggerated. It's maybe the consciousness of a politician. It fits with that. But the potential for that is enormous, because we might be able to jump into something more in terms of the computation that provides the self-organization of tissues and what can we manipulate. So I do think that this opens the door for a new dimension in our area. And just to end up, another thing that we've been exploring successfully over the last years, now we are ending up the experiments, is the idea that collective intelligence can be translated into living things by engineering living things. Collective robotics is something that has been there. Collective sum intelligence has been used by different people, different contexts, to imitate the way you approach ants and bees and other social insects. Again, is one of the big questions. So how use sociality, collective intelligence emerge in evolution? And we can approach that from theory, from the observation of nature. We can build robots, like this one, which can be programmed. It's already an old one. It can be programmed, has sensors, has a clock that was, of course, built in Switzerland. And now we can do something else. So you can manipulate living things to behave like robots. Or, much better than that, at some point we realized that some of the mathematics we were using to model some of our constructs in synthetic biology were very, very close to things that we have been using in many other people, in modeling the way, for example, ant colonies make decisions. Because again, they can be seen as multi-agent systems that interact with each other and perform collective computations. And by realizing that, we realized also that it was possible to actually engineer, for example, Escherichia coli cells, microbes, in such a way that they behave like robots, little robots that behave like ants. So now in the final phase of modeling that, building that in the lab, we have our E. coli cells that behave like ants and that respond to the stimulus by others in ways that do not exist in nature, that imitate ants, and that...
Can you explain how this is imitating an ant?
Well, this in the simplest experiments that can be built, for example, with ants, you put an ant nest in one place. Let me show you this. In one place, in the other place you put food, okay, and you have two branches. One is shorter than the other, and the thing is how the ant colony detects the shortest path, right? Single ants cannot do it, but ants initially they go randomly through the two branches. When they find food, they go back and leave a signal, chemical signal, a pheromone, and if other ants find out that signal, they tend to follow it and reinforce it. So if the signal is left in the longest part, it also evaporates easier because you have more space to evaporate. So what happens is that by this reinforcement mechanism, collectively the system is able to solve the shortest path problem, which is the simplest one that we can formulate, but many others. So essentially what we do is to build a system reusing parts that are there, where essentially they leave a chemical mark and react to the mark in a way that the others can follow.
So the E. coli is engineered to also leave marks in the environment.
Sort of, yes.
What do you mean, sort of?
Well, because they cannot leave a mark that is on such a solid medium like that. It diffuses, but essentially it's the same idea.
Sure, but now the E. coli that's the innovation. Mm-hmm. The E. coli is sort of also excreting something.
Yes.
In the environment continuously or transiently.
Transiently, I mean, it responds adapting to what is being detected in the medium.
But for instance, the E. coli is only releasing something when it detects, let's say, some sugar or something, or
some threshold of a chemical signal, yes, which is called a lactone. But it's
okay. But then the E. coli is not necessarily going back to a home base because it
No.
doesn't have a nest. So
Not necessarily. No.
So how do you define that in a navigation task?
Okay. If you go into the simplest solution, then they just go to one place, and by a mechanism of symmetry breaking, you have more concentration in one side of the. I don't have the pictures here. One side of the arena, if you want, of the dish, and the others reinforce that and get accumulated into one place. But essentially, you will see a mark that goes only in one direction and is followed by others. Then you can reformulate that with microfluidics, but it's a longer story. So you can make the go back to the place possible, which is part of the thing that we are doing now.
Okay.
Right? And so the dream is to be able to manipulate that. There's a big number of reasons for that. It opens the door for a decision-making engineering that connects with pattern formation, connects with collective behavior, and connects in terms of beyond the theory that we want to understand, connects with a number of processes that happen inside our bodies also. The way your bones are built and maintained has to do with a collective behavior of two different types of cells. When this collective behavior fails again, you have diseases involved in bone. Just the end picture. By analyzing different systems and trying to make kind of a global logic of what computation means in nature and what kinds of computations do we have. Again, connecting with the original question, what things do we observe, what things we don't observe, and why is that? We made this what we call a morphospace of potential forms of computation. I'm sure that this is going to change over the years because it's just a qualitative picture. Where I put in the axis here is the diversity of agents. How many different kinds of agents are there? From a single type of cell or single type of ant to a diverse collection of cells. The spatial embedding, how important is space? In most contexts, a well-mixed thing is okay, but in reality, most of the time, space is there, is relevant, and it impacts computations. And in the other axis, which is probably the most complicated one, I put parallelism, which is it's a tricky thing because our distributed computational thing can be called parallelism, but can be called other ways. And essentially amounts about how much sequentiality is embedded into the system. So I put a number of systems that you can identify in natural systems. The size of the spheres as a tentative sizing of how many examples do we know of that. And we think that distributed computation, the way we have found it, occupies a domain in this space that is largely unexplored. And what I just show you these microbrains that can be built, I do think might define a new dimension in this perspective.
Now, for instance, in this case, you are placing DNA computing and neural systems in a specific way, only separated on one dimension, apparently. So can you explain that? Why you see the separation only along that dimension?
Let me see if I remember that.
Just invent
The DNA computer it's quite free from space. It's essentially mixed, except for the last examples that have been done on chips. But
wait, in the DNA case, the conformation would be rather important. So there is a spatial dimension.
Well, at that scale, at the agent scale, if you want.
Yeah.
Yeah, but the arrangement in space of the parts is not relevant. You can think the well-mixed case. Agent diversity is extremely high. It's quite high because you have diversity of nucleotide sequences plays a leading role here. The potential for combinatorics, and of course, it's highly parallel. Neural systems is a tricky one because what do you mean by that? Of course, it's parallel. There's a diversity of agents. A diversity of cells in the context that you do have because the learning process that if you want to characterize every single element in the system, the way it connects with the others defines a diversity there. Okay, and of course, embedding is important.
But that might be true for DNA as well. right? Because in the end, you have complex regulatory networks that implicitly would define relationships among different genes and how they're expressed.
But this, I would say, more than regulatory networks, I would say more like combinatorial chemistry, more than that. I would put that more into that place.
Okay.
It's a tricky thing.
Understand this space.
Yeah, well, every time I show that, it's people pointing to different places, saying, "Well, I'm not completely agreeing on that." It's a kind of tentative space. Our main question is: natural computation occupies, if you build a morphospace, occupies what part of it, and why is that? And to what extent engineering can push you outside from what is being occupied by nature.
Right.
That would be the ambition of that. So I think that's all. If someone of you is interested in knows to read Spanish and interested in this kind of big perspective, I had this book that was published last year, Synthetic Lives, which I hope to be translated in the future, has a very modest subtitle, which is an approximation, a revolutionary approximation, to science, history, and the mind. So I think that you should buy it.
Yeah, that's very humble.
Just in case.
Humble.
Very humble,
Yeah.
very humble, yeah. Yeah. My mother could have suggested that.
Exactly.
Thank you very much.
Great. All right, question time. Tony.
Thank you, very great talk. And
Maybe you want to show your evolution T-shirt because to impress Ricard.
T-shirt.
Yeah.
Evolution is a bicycle. The idea that you could reprogram cells to create a new network that makes insulin is a fantastic idea. But why don't you just go directly for the autoimmune disorder and stop the body from destroying its own cells? Is that not possible?
Because we don't know how to do it.
Right. Why is that such a complicated problem?
Why? So what is the question?
Why is it so difficult?
Why is it so hard to understand autoimmune disorder?
Well, think about there's something that theory allows to understand.
Right.
The immune system is an amazingly effective system because it builds inside it by an extreme combinatorial capacity of preparing you into potential invaders. Essentially, you have a machinery that, for billions of potential invaders, they can target them. But what happened then? What happened is that it's inevitable. You can calculate that if you optimize the system to do that with the resources that you have. There's a little possibility that in your lifetime, something that is a target inside your body, a piece of a protein, for example, from your bones or the cartilage, are recognized as alien. And the immune system tries to remove that from you, which is a problem because it's yours. So how you teach your immune system that this is wrong? Well, that's a big problem. You cannot.
What do you mean you cannot?
Well, so far, how do you teach the immune system that the cells that are already building the response against your cartilage, how do you tell them that they don't have to do it?
Well, they have developed an affinity for aspects of cartilage. So you could imagine that you invent some magical substance that sort of binds to those sites on these antibodies, and blocking them from actually attacking your cartilage.
Yeah, go to the people who's working on that and tell them
No,
that.
wait, we're challenging you, right? Because you want to go one. I posed a similar question. That you say it's like, oh, this is like a computational problem, and I have to build this controller, and I have to move these cells away, and just produce them in secret somewhere else underneath the radar of the immune system, right? And Tony is sort of teaching you in the same way. We say, yeah, but it's a bit of a detour.
Well, it depends. If the autoimmune disease is something that you can solve by bypassing the fact that there are target cells that are being destroyed, and the function of those cells is very specific, and you can rebuild the function in another type of cell that is not being targeted, then you have a solution, a potential solution. For the cartilage of the bones, for example, for arthritis and things like that, it's still a problem. We don't know. I always tell my students of bioengineering, the important lesson, I think, is that we don't know, but I'm very convinced that there might be ways of solving the problem. In a way, the system is also falling on a tractor that identifies part of your body as an alien part. Okay.
Well, I think the solution you propose is a nice, potentially nice way around the problem. But what I worry about is you introduce a new circuit into the body that the body is not prepared for. How does it regulate that system? I mean, you could just uncontrollably produce insulin or whatever.
Well, you have to put the that's why it's a circuit. It's not just a sensor that detects something and releases something. You have to put a circuit that replaces the whole machinery. That's the big challenge. If it was just releasing insulin from a given cell, that's doable now.
So it's something you're saying you want to emulate the pancreas using other cell types.
Kind of, yeah.
Right?
Kind
That's
of,
roughly
yeah.
what you're saying.
Yeah. But instead of having cells in the pancreas, maybe having muscle cells and then something in the immune system that plays the same role and bypasses the problem.
But do you plan to grow these cells and then implant them, or they will just develop inside?
We'll see. We'll see. I wish I would have the. Right now, we are in the process of building the circuits in cell cultures and see how far can we go with that. And in the meantime, having a discussion with the people in the clinics to see how feasible it is to translate that into human cells and see what happens.
Vicky?
So I keep creating such cells for, let's say, medical purposes, but from what I got, you could actually use this kind of approach to create, let's say, a computational circuit. Is that correct?
I don't think I heard.
Vicky, it's nice you sit all the way in the back, but you have to adjust your voice to that distance.
So I say that I get using these circuits for medical reasons, but from what I understood, you could actually use this sort of approach, let's say, for creating computational circuits that you can use for whatever sort of, let's say, mathematical or more mechanical approach. Is that correct?
No. Well, no. I mean, it would be unfair to say that you misunderstood. I think my talk gives some insight into that direction. On the one hand, we as theoreticians, we want to understand computation in natural systems. And we ask ourselves this question. I mean, could it be possible that maybe part of what we see in natural systems doesn't fit well into the standard, if you want, engineered or mathematical approach to computation? That's what I think that the point that was being raised here. There's a lot of the things that happen inside cells that we don't understand. Usually, when I show these nice movies of DNA unfolded and proteins like that, this is very, very far from what's happening inside cells. It's extremely crowded, extremely noisy, and we are still wondering, to a large extent, how is this possible but this works, and works in such a reliable way. And lots of things that we assume, like we know that you target a gene, you silence the gene, or you remove it, and the function performed by that gene is performed by someone else that was not doing that before. You will say, of course, it's a redundancy. But it's not redundancy. It's something else. We are quite far from understanding that. So as part of our ambition in terms of thinking in computation is what makes computations different somehow in biology with respect to standard computers. But not because we want to build a computer that competes with any machine.
Please.
Okay, well, actually my question was pretty similar because you said that you could manipulate cells to act as logic gates, right? And like computers are just like a big system of logic gates. So I thought that this could mean that you could build like a custom computer made out of organic tissue. But you were saying no, partly because of the noise.
It wouldn't be interesting, I think.
Yes, because I think that would lead to brain prosthetics. But is it so far?
Yeah, it's quite far from that. To be honest, I would love to say the opposite, but
you just have to study, okay? It has to come from the books. All right? Not from Ricard with some mutated cell. Okay, Giovanni.
So thank you for a great talk. So, I have a more general question on the paradigm that seems to me that you use. So I would say that most people doing evolutionary computation or emergent computation, they really make very few assumptions on what are the components of the systems. They just use the evolutionary algorithms to bootstrap the whole system. Whereas it seems to me that your approach is slightly different, because in your approach, such as in the cells, some components are really engineered, and then it is just the interaction that you leave completely free. You leave. That's at least what seems to me. So the interactions are not hand-wired, but they are maybe emerging. They are. So it seems to me that the paradigm is a bit different, because in a sense you have components that really resemble control engineered systems, but it is their interaction that you are really letting free or letting emerge. Is that?
It's more like we know that one of the big problems is the wiring. So the wiring is the origin of most of the problems of synthetic biology. And by avoiding the wiring, which is what you really want to do, is we ended up in making the system more modular, more able to be reused in different ways. But we discovered something else, that we can go into something that is not standard design. So you break into pieces something and you distribute the computations between subcircuits. And that has a lot of advantages for us. And that is not the standard metaphor. So it was in trying to go into simple circuits, we find out a new design principle.
Tony.
Yeah, that relates a bit to what I was thinking. So artificial life in the 80s and 90s had this big explosion. And then it kind of died down a bit because people discovered that evolutionary algorithms couldn't really solve all the problems that were really tough. It was the same problem that we'd had before of very large search spaces, and you could only explore small parts of them. And then there was this notion that we have to understand the evolution of evolvability, that you can only solve complex problems by having complex systems, and you want to start from somewhere that's not tabula rasa. You want to start with a complex architecture that can evolve in that direction. So, I mean, are you sympathetic to that view? And if so, what is the state of the art in terms of understanding evolvability and having these more complex architectures that we can use to explore design space?
I think that it's an interesting time now for the problem of evolvability. It's something we have been exploring ourselves, especially because what people is finding when you have the potential for manipulating regulatory networks, you can ask, for example, to what extent I can randomize interactions between genes and see if this still works. And one of the interesting findings in the last, there was a paper in Nature 3 years ago by colleagues in my institution, that you can randomize a lot of the wiring diagram in E. coli in particular, of the regulatory network, and the system keeps working. So, which again is kind of surprising, thinking that you have been fine-tuning things, especially in microbes, you would say. You have to not to waste things, and you have to do things kind of optimized way. But nevertheless, there's a lot of potential for rewiring, which people take like one kind of signature of highly evolvable network. But the principles that pervade that, why is that, and what does it mean that? We are still far from understanding.
Stefano.
So you're building those microcircuits that are functioning well if you are somehow outside of the body, because you restrict the number of proteins to be the one that the microcircuit is interested in. But there is no evidence that this microcircuit will behave the same way when you put it inside the body where there are all the other proteins. So somehow the computation of these microcircuits is defined by the surrounding space. And
yeah, you're raising an important point. I mean, building that in a cell dish, in a cell culture is certainly one thing. Building something that has to be in the context of the body, close to a particular niche, might be a challenge. So one of the things that we are quite hardly exploring is how to make the circuit as robust as possible. So kind of a niche independent. I'm mentioning this because, for example, in other contexts, in gene therapy, for example, there was the promise that you put back cells of people having Parkinson in the right place, and that will regenerate the tissue that is being destroyed. And what happened, at least in the first experiment that people did, was that the cells that you put there were destroyed, and that was all. And the thing that was, people realized that you have to put the cells that have to do the function is in the substantia nigra, which is this particular target, but also the cellular context. So the glia cells that are there that replenish and not nourish the cells, and other things. So the niche is important. And you're completely right. I mean, inside our body, it's not like a playground. You have to put things into the right context. For that particular case of diabetes, we have transgenic mice that will play the role, the intermediate role before going to humans, which are quite good designs for testing that. So if that works in the mice, it should work in humans.
Yes, please.
Trying to implement switches in your yeast library. Did you ever consider to exploit the capability of yeast to be in both diploid and haploid form, affecting the stoichiometry of genes involved in computation?
No. No, we didn't. We avoid that, as we avoid other things that were kind of a burden, because we wanted to make a proof of concept, something that could be replicated. But yeah, but considering the haploid diploid, yeah, I mean, I guess that we haven't thought about it, but maybe it's something interesting to consider. In a way, it's information, and it's potential redundancy or not. Who knows? Think about it.
Yeah, so if we now look at all these examples, it also gives a bit this feeling of FM at work, right?
FM?
Yeah, fucking magic.
Oh.
So the point is that how much of the system actually really understand that we can engineer it, right? Because sometimes you have small examples, but as you say, there's an enormous amount of redundancy in these systems, right? So how much of the system that you're working on, even for an E. coli, for instance, is at this point in time really sufficiently understood to declare it an engineering target? And so where are we actually really in this whole adventure?
Yeah, if you're asking for our understanding of the regulatory pathways or the whole machinery, we're quite far. And the funny thing is that even for the supposedly simplest microbes that have been studied, like people in my center that uses this Mycoplasma genitalium, which is supposed to be very simple microbe, when they completely mapped everything that they could, the whole picture is quite much complicated than we even suspected. Mm-hmm. So the good news, I think, is that we do not pretend, and I personally think we are not ever going to do it, mm-hmm. To act on that scale. But there's some space mm-hmm. available in engineering parts of the system. Mm-hmm. And the whole cell is something that carries our computations, mm-hmm. Hopefully safely somewhere. Right.
Now, but there's an interesting implication, right? Because then you could argue, well, maybe this notion of computation you started out with is actually not really giving you leverage to understand and engineer this system. Maybe the starting point of computation is actually not working, and maybe we have to rethink that. How about that?
Yeah. I unfortunately have to agree.
Okay.
Yeah.
Hmm?
Yeah.
No, but this will be really important. So if you look at, if we now generalize it towards brain sciences, often you feel, okay, we're in this straitjacket of the Turing machine, which is actually really limiting our view. And it's something you observe something similar, right? That, yes, there are incredible advances, but on the other hand, compared to the complexity out there, we're just scratching the surface, right?
Yeah. Yes. And as I was saying, there's a number of things that we already know that happen inside cells. This redundancy metaphor that we know now that it's just just explains maybe a 10% of the non-effect of removing genes.
Mm-hmm.
There's a huge amount of things that are replaced by the system when we should expect to see a failure.
Right.
And that's what we call degeneracy or distributed redundancy, or distributed robustness. But the origin of that, we still don't know. We played a lot actually by evolving circuits. And I remember some years ago that we had these whole maps with drawings and trying to figure out what was the logic of that? What is the origins of that? And I think that's something that escaped. This nested robustness escapes from being encapsulated in a simple theory, or in a theory right now.
Very good. Do we have other questions? Yes, please.
There is the whole problem of the real origin of life in the Earth. All these of the artificial life have some contribution to this discussion.
Yeah. Well, many of them. And, well, the origin of life is still an open question. But one of the interesting things of the advance in artificial life that has been moving from simulation to the wet lab. That's something I haven't mentioned, but we have been involved for years in modeling protocells. So what do you need for now a physical cell to grow and split? It might sound simple, I know. But I remember it was maybe 7 years ago when we started the European project that the target of the project was to build an artificial cell from scratch. So you know that we failed because we didn't get the Nobel Prize. But it was interesting to see that at the beginning of the project we had the theoretical part in our side. And I thought, well, probably someone has already published a paper on a physical model of a vesicle that grows and splits. To my surprise, no one had done that. And because even that is quite difficult. You go against the physics of building symmetric things that are very stable. And I think the area has provided, over the last 10 years, a lot of advance to that. And my bet is that in the next 5 years, maybe, some lab will build an artificial cell, a real artificial cell from chemistry. So because we are closer to the principles.
But that you're saying that the Ventnor example you don't buy yet.
No. That's a genome transplant, okay, right. If you want.
But it's important to see that, right?
Yeah,
because
yeah.
I think it's a bit over-interpreted.
It is over-interpreted. It's again a sociology thing.
Right, exactly.
It's actually funny because if you look at the first interviews with Ventnor, he was very cautious, like saying, well, this is not really. But then journalists become wild, and then he did too. Right, exactly. So it was like...
Exactly. But I thought for you guys, life on Earth came from the Santa Fe Institute landing on this planet, and then life emerged. Wasn't it like that?
Yeah.
Exactly.
Yeah, of course.
It's solved. All right, so let's have the coffee break. Thank you.